Biology 9700, paper 21, May, June 2024. Question number 1 to question number 3, part 1. So, question number 1 is figure 1.1 is a diagram showing part of the cell surface membrane of an animal cell. So, you can see it is labeled with A, B, C, D, E, F, G, H. State the approximate thickness of the membrane as shown by the line G and to H. So, you know, students, that the uh, thickness of cell surface membrane in case of animal cells is about 7 nanometer. So, compare table 1.1 to show the names and functions of the components of the cell surface membrane, the letters of the labels 1.1 that identify each component. So first of all is the channel protein, and channel protein is labeled with the letter A. So the function of this channel protein is to facilitate diffusion, or the movement of ions. And phospholipids are with the letter E, and the function of these phospholipid molecules are they form a bilayer so receptor for signaling. So they are the glycoprotein or glyco glycoprotein, and with the letter they are shown is B, and the next part, which is F, and F part is a cholesterol molecule, which maintains fluidity. So these were the different parts labeled, the name of all these parts. Now we move towards the next question. And next question is figure 1.2 is a drawing of transmission electron micrograph of a cell from the palisade mesophyll of a leaf. The drawing does not show all of the organelles visible in a transmission electron micrograph. So you can see complete figure 1.2 by drawing and labeling a mitochondrion, rough endoplasmic reticulum, and smooth endoplasmic reticulum. So your drawing should show the details that can be seen in a transmission electron micrograph, and then identify the organelle labeled X and state one function of this organelle. First of all, and the function of this organelle. So here, first of all, we are going to draw one mitochondrion, and you can say the mitochondrions are of rod shaped. So they are rod shaped and double membrane structure, and crystals are there. And you can say the endoplasmic reticulum, they originate from the nuclear membrane. So in this way, they name cisterns. And if the on them, you can say in this way, they form cisterns. So if ribosomes are present on them, so they are rough endoplasmic reticulum. And if they are without ribosomes, they are smooth endoplasmic reticulum. So in this way, we have drawn all these parts. Now we are moving towards the next part of this question. This is identify the ordinally labeled X. The ordinary labeled X is the Golgi body, and the function of Golgi body is the modification of the protein which was stored into it. Modification and packing of proteins. It modifies the protein which is present in it. Next question number 2 is there, and you can see water is a main component of blood. Explain how the properties of water make it suitable as the main component of blood. You know, the blood is a tissue which has to move from one part of the body to the other. It passes through the heart. It is pumped by the heart, and then in this way, it provides the oxygen to all parts of the body and collect the carbon dioxide from all parts. So in this way, blood is very important and main component. So because it is very, you can say, important part, so it is its first property which can be written as, that is the water is a good solvent. A solvent which dissolve many and can dissolve many substances into it. And the second property which can be written as the, it transports the many substance such as urea, hormones, etc. And the number 3, we can say it maintains the temperature of the blood. Or regulates the temperature. Okay, next question is, a picture has been shown on the screen. 211 is a diagram of the circulation in a mammal. So P and Q, these are the two labelings. And first of all, we are moving towards the question. Complete figure 2.1 by naming P and Q, which write your answers. Okay, here you can see P. You can see the blood from body is entering into the right atrium. So it means this is the vena cava, which is bringing the deoxygenated blood after collecting from different parts of the body, and then it throwing this deoxygenated blood into the right atrium. And on the other hand, the P, it is receiving the blood from the heart. So the blood from heart is moving towards the different parts of the body for distribution. It means this is aorta. And you can see the vena cava carry deoxygenated blood, aorta carries the oxygenated blood. Vena cava collects from different parts and then it takes towards the heart and into the right atrium. And whereas from the left ventricle, the blood which is oxygenated, and then this blood moves towards the other parts. So So aorta and vena cava, these are the two main parts. Now describe the function of these. Already told you that the vena cava, P is vena cava, and the Q is the uh, yes, P is aorta and Q is the vena. So aorta supply the blood from oxygenated blood from heart towards body parts, and more pressure is required for distribution. More pressure is required. Whereas the vena cava collects and brings the blood, deoxygenated blood, from body parts and bring it to heart. Explain why the mammalian circulation is described as closed double circulation. Because it is called as closed because it is, you can say, uh, it is circulated through vessels. Through vessels, so it is a closed. So it is not flowing in the body without vessels. So it flows through vessels. It is circulates through vessels, blood vessels, three different type of vessels. So that is, it is called as the closed double circulation. So the double circulation, it passes through the heart twice, once in the form of oxygenated blood, the other in the form of deoxygenated blood. So there are the two circulations of blood in this way, that from between pulmonary and systemic. And pulmonary is between heart and lungs. And systemic circulation is between heart and body. So in this way, pulmonary circulation, the pulmonary artery takes the deoxygenated blood from the heart towards the lungs, and from lungs, the oxygenated blood is carried by pulmonary vein towards the heart, into the left side of the heart or the left atrium. Whereas the systemic circulation, what happens? The deoxygenated blood is brought to the heart by vena cava, and then the oxygenated blood from the heart is distributed to other parts of the body by aorta. So in this way, the blood has to pass through the heart twicely. So it is called as double circulation, and it is closed because it circulates through the blood vessels. Now the next question is, you can see our diagram over here. Figure 2.1 is a transmission electron micrograph of a cross section of an arteriole. Blood flows from muscular arteries through arterioles into capillary networks. The lining of the arterioles is folded because the arteriole has constricted. This constriction causes the blood pressure to decrease from 12.7 kPa in the muscular artery to 2.7 kPa at the end of the arteriole. Now you can see the picture. Now we are moving towards the question. Explain why it is important that the pressure of blood decreases as it passes through the arterioles. From aorta, the blood flows into the arteries, and from arteries, it flows through the arterioles. You know, the aorta has high pressure when it moves towards the arteries, and the from arterioles, it is slowed down as the diameter is decreasing from aorta to arteries and to the arterioles. The diameter is decreasing, so the blood has to move great pressure. So at this case, if the blood reaches to the arterioles, a little bit pressure is reduced. It needs to be reduced because in this way, if there is high pressure, the blood vessels can burst due to high pressure. Vessels can burst. To avoid their burst, so it is, you can say, widened to have high pressure. And secondly, the capillaries have thin walls. Thin walls. And secondly, exchange of gases has to take place in the capillary. Has to take place. So this is the reason the blood pressure is reduced over here. Compare the structure of the muscular artery with the structure of the arteriole shown in the figure 2.2. So here you can see that you can say if we want to compare, first of all, we have to discuss what are the similarities. What can be the similarity between these two types of blood vessels? You can say both of these blood vessels have endothelium, internal lining, endothelium, internal lining. So both of these blood vessels have these internal linings. Secondly, they have smooth muscles. And as the question is of 3 marks, so we need one more, you can say we can write a difference. So the wider lumen, you can see the arteriole is muscular, and this, you can say this muscular, all they have the muscles inside, but the difference of the lumen is there. This arteriole has wider lumen as comparison to muscular artery. Now, question number 3 is there. A class, uh, a class of students was studying the features of some human pathogens. One of the students constructed a flow chart to identify 4 different human pathogens. The students used information about the structure and mode of transmission of each of these pathogens. So we are moving these flow charts. Complete the flow chart in figure 3.1 by identifying the mode of transmission. We have to write the mode of transmission, and then we have to write the scientific name of the pathogens. So the name of one of the disease. So name of one of the disease, we have to complete all these in these. So first of all, the first they have done for us, that is pathogen has a cellular structure. If we go on this side, no, pathogen is transmitted by fluid. So it is transmitted by fluid. So it is HIV, human immune virus, and the disease which is caused by human immune virus is the AIDS, acquired immune deficiency syndrome. So in this way, the pathogen which has a cellular structure, but no cell, it has don't have the cellular structure, but pathogen is transmitted in body fluid. So HIV is caused the AIDS. On the other side, if we go, yes, the pathogen is a prokaryote, and pathogen is transmitted by fecal oral route. So in this way, the contamination This is the disease which is caused by the contaminated food and feces. So it can be the vibrio cholera, and the disease which is caused is cholera. On the other side, pathogen is a prokaryote. No, pathogen is transmitted by vector, and the disease plasmodium is the cause of malaria. The last one is pathogen is transmitted by fecal oral route. No, pathogen is transmitted by air droplets. And the tuberculosis, the disease name is written. So Mycobacterium tuberculosis is the name of. So we have completed this flow chart in which the different disease has been shown. Now we are moving towards the next question. Here you can see HIV has a nucleic acid core of RNA. The virus also contains the enzyme reverse transcriptase. After HIV entered T lymphocyte, reverse transcriptase catalyzes the formation of DNA using activated DNA nucleotides with the viral RNA as a template. And you know, in reverse transcription, there is formation of DNA when RNA act as a template. Some drugs such as tenofovir have been developed to inhibit the action of reverse transcriptase. It means tenofovir act as a reverse inhibitor. The structure of the tenofovir is similar to the structure of deoxyribose adenosine monophosphate. Deoxyribose adenosine monophosphate. The AMP. The structure of fur is similar to the structure of, so you can see the structure of tenofovir and deoxyadenosine monophosphate. So you can see their structures. Now we will move towards the question which they have asked. So the first question related this picture is, after tenofovir is absorbed into the cell, it is phosphorylated twice and can be used by reverse transcriptase in the synthesis of DNA. So when a tenofovir molecule is added to the DNA strand being synthesized, the process stops. Suggests the mechanism of action of tenofovir to prevent the synthesis of DNA by reverse transcriptase. Use the information in your. So suggest the mechanism of action of tenofovir to prevent the synthesis of DNA. So here you can see tenofovir will competes, and then as a result of this competition, it will prevent the nucleotides being added to elongating chain. So it will compete tenofovir will compete with active sites and stop, inhibit the reaction by acting as a inhibitor. And so it will also make a phosphodiester bond. Phosphodiester bond, and it will stop the further formation of diester bonds. So further formation of bonds will stop and the reaction. Other words, you can say the reaction will stop. Now the question number C is part C. Pre-exposure prophylaxis is the use of therapeutic drugs to prevent the replication of HIV in the body following infection. The drugs are taken by people who are at risk of becoming infected. Tenofovir is one of these therapeutic drugs. In 2016, the United Nations UN set a global target of 3 million PrEP users by 2020. Table 3.1 shows the number of people uh, across the world who received a therapeutic drug for PrEP in each of the year between 2012 to 2019. So 2012 it was 10,000. Now in 2019 it was 605,000. Now we have to calculate the percentage of people who received PrEP in 2019 as a percentage of the target set by UN in 2016. So the target which was set up at that time was it was 3 million. So we will write calculate the so the it was in 2019 it was 605,000, but but from the target which they had set was 3 million. So we have to find out the percentage in this way. So it will be 20%. The answer will be 20%. Next question is state and explain how health authorities can reduce the transmission of HIV. So transmission of HIV can be reduced by different methods. First of all, by using condoms, which can reduce the transfer of uh, virus from one organism to the other. So blood transfusion, for blood transfusion, use of disposable syringes and sterilized instruments in use of different intrauterine devices, for example diaphragm or coil. So and the other you can say the uh, different drugs for the safety of people. Sharing of needles will prohibited. Will be prohibited. So it was all about the question number 1 to question number 3. Please students like, share and subscribe my channel and thanks for watching.